Replace an ill kidney with that of a pig? Yes, they have done it with the heart as well, not just the kidney. Scare mosquito fish off with a robot fish? Yep, that's been done. And if you want to learn how worms regenerate, you are recommended to make them glow in the dark first. Welcome to Give a Fact, and you are watching Bizarre Genetically Modified Animals. If you like this kind of video, be sure to subscribe for more great content, or tell us what you want to learn more about in the comment section below. Now, let's get straight to the video, shall we? 1. Make animals glow in the dark. Adding fluorescent genes to animals' DNA has been done since the beginning of 2000, but it never ceases to amaze us. In 2021, a team of researchers led by a professor from the Organism and Evolutionary Biology Department at Harvard has taken the study of 3-banded panther worms, which can regenerate themselves when cut off, to the next level by making them glow in the dark. The glow-in-the-dark factor comes from the introduction of a gene that, when it becomes a protein, gives off certain fluorescent glows. These fluorescent proteins can either glow green or red and can lead to glowing muscle cells or glowing skin cells, for example. The ability to see the details of how the cells work and how they regenerate will give the scientists information and clearer ideas of its mechanism. Researchers can do very precise manipulations in the case of 3-banded panther worms, such as switching off certain genes to force it to make mistakes when regenerating, like growing a tail instead of a head, or 2 heads instead of 1, and in the wrong place. This can ultimately help scientists truly narrow down what genes are required for the worm to carry out its usually perfect whole-body regeneration. Now, with the ability to make transgenic or genetically modified worms, the researchers say they are most excited to study a population of stem cells critical to regeneration. The cells are called neoblasts and are believed to be pluripotent, meaning they can produce any other cell type in the animal, such as neurons, skin cells, muscle cells, or gut cells. 2. Web-spinning goats. The dragline silk fibers that spiders manufacture is among the strongest material in nature, and 5 times stronger by weight than steel. If sufficient quantities of spider silk were available, it could have potential uses such as medical sutures and biodegradable fishing lines. With this in mind, scientists attempted to farm silk spiders, but they failed on any scale because of the spider's cannibalism habitat. In 2000, Nexia, a Canadian biotechnology company, found a new solution: a goat whose milk contained a spider protein for web making. The scientists inserted that spider dragline silk gene into the goat's DNA in a way that would only affect their milk. As a result, this genetically modified goat's milk became less drinkable and much more of a silky substance called the silk milk. This was said to be used for the manufacturing of BioSteel, a web-like material by the company. So, did that research ever lead anywhere? And what happened to those goats? It all went a bit quiet because Nexia Biotechnologies, the mother company then, swiftly went bust and sold its 2 genetically modified goats, Sugar and Spice, to the Canada Agriculture Museum. Here, the goats were displayed until 2013 due to public pressure. Creating high-quality and durable products for human use from this mind-blowing science technology is surely no easy task. So even now, more than 15 years later, Dr. Randy Lewis of Utah State University and his team are still looking after genetically modified goats for silky milk research. Although he doesn't know where the first 2 goats, Sugar and Spice, have gone, his lab is now home to over 20 web-spinning goats. This project, of course, has been quite controversial because of many concerns about the health of the GM goats. Dr. Lewis said that in over 15 years of breeding these goats, we have no indications of any abnormal health risks. We have simply transferred a gene for spider silk protein into the goat genome and have put it under the same cell regulation system as the other milk proteins. So it is only made during lactation and only in the udder. All are compliant with regulations, he says. Right now, both the USDA and the FDA are regulating the transgenic animals, and since we're not beyond the research stage, they have not required anything beyond their standard transgenic animal regulations. This project has caught the interest of Inspidair, a biotech company in the Netherlands which has been working on bulletproof silk skin, and a manure-derived bioplastic called Mestic. 3. Successful surgical experiments of pig-to-human organ transplant. In the United States, over 100,000 people are on the national transplant waiting list, about 90% of whom need a kidney. But in 2019, less than 40,000 transplants occurred. About 17 people die each day while waiting for an organ, according to the Health Resources and Services Administration. Scientists have long sought to solve this shortage by using animal organs, a field known as xenotransplantation. Pigs have emerged as the primary focus of this research, since their organs are anatomically similar to human organs, and the animals can be bred in a highly controlled manner. When researchers tried such experiments using non-human primates in the mid-20th century, the scientists found that simply transplanting another species' organ into a person causes the immune system to revolt at the presence of a large foreign object. In recent decades, genetic engineering has overcome some of these challenges. Scientists learned that the aggressive immune response seen after a pig xenotransplant is spurred on by antibodies that detect a specific sugar molecule called alpha-gal that dots pigs' blood vessels. It's the same sugar molecule responsible for some allergic reactions to red meat. Then, in the early 2000s, they devised ways of disabling the pig gene responsible for this sugar. In 2021, surgeons in New York City successfully attached a pig kidney to a brain-dead human patient, but still alive on a ventilator. Within minutes after the surgery, the kidney started producing large amounts of urine and showed other signs of normal functioning. The team kept close tabs on the kidney for 54 hours and saw no signs of rejection. This is the first time that a pig kidney has been transplanted into a human body and has not been immediately rejected, marking progress toward the goal of drastically expanding the supply of life-saving organs. Less than 2 months later, the same surgical team repeated the experiment. A third such transplant from researchers at the University of Alabama at Birmingham, this time into the abdomen of a man kept alive temporarily by a ventilator following a motorcycle accident. The first pig-to-human heart transplant happened in a living patient who was suffering from acute heart failure in January 2022. He survived for 2 months with a pig heart before dying of heart failure. Then, in July 2022, 2 pig hearts were successfully transplanted into brain-dead people with heart problems. The team monitored both patients for 72 hours before taking them off life support. For those 3 days, the hearts kept the recently deceased patient's blood flowing. However, the new heart was too small for the patient's chest, so surgeons had to adjust blood vessels to account for the size mismatch, and blood flow wasn't perfect. Before pig-grown organs can go mainstream, researchers will have to demonstrate that the organs can survive attacks from other players in the immune system and last for months or years in the human body. For example, over time, T cells, a type of white blood cell, can come to recognize the transplanted organ as foreign and attack. Immunosuppressing drugs can help mitigate these sorts of responses, though the side effects of such drugs, such as susceptibility to infectious diseases, can be a major burden. Pig organs can contain retroviruses that lie dormant in genes. So, how about the ethical side of the story? Performing the first of its kind operation like this on a living person raises many ethical questions. A future where potentially millions of pigs are raised to harvest their organs for humans raises significant ethical questions. The organization People for the Ethical Treatment of Animals, or PETA, said in a statement that pigs weren't spare parts and should never be used as such just because humans are too self-centered to donate their bodies to patients desperate for organ transplants. Advocates for xenotransplantation argue the potential benefits of vastly expanding the organ supply are worthy of any potential harm done to pigs. Nearly half of the patients waiting for a transplant become too sick or die before receiving one, said Montgomery, the NYU surgeon. The traditional paradigm that someone has to die for someone else to live is never going to keep up with the ever-increasing incidence of organ failure. For now, pig-to-human transplantation has logistical issues that will help restrict its use. For one, the painstakingly engineered pigs are hard to come by. In addition to their rare genetic pedigree, they must be raised in sterile environments so they don't pick up any microbes that might be transferred to humans. Another possible hurdle: animal rights groups have decried the idea of pigs as spare parts for humans as cruel and dangerous, a sentiment that society will need to square with the urgent medical need. But first, doctors need to know if xenotransplantation is even possible or will once again fail. And that is a wrap. What did you learn from this video? Let us know in the comment section below. We hope that our educational videos will help spread the knowledge so everyone can protect our world and biodiversity together. Please like and subscribe to our channel if you find our videos useful and want us to make more videos. Until next time, give a fact.